All right. So, hello everyone. A very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for today's webinar on microbiome in health and wellness hackathon. You have probably heard the term hackathon before, but unless you are, let's say, already involved in a tech industry, you're likely wondering what is a hackathon exactly. It sounds so trendy and exciting, but what does it actually entail? A hackathon is often a competition style event that brings together individuals to develop new approaches to solve problems or complete large projects that require collaboration. And one of the best things about participating in hackathon is the opportunity to meet new people who care about the issue or technology that you care about. Whether it's for project collaboration, finding a mentor, or even potential employers, hackathons are a great place to make connections that could pay dividends in the future. And that is why this November we are organizing a hackathon on microbiome in health and wellness to provide students with a platform to showcase their innovative mindset of problem solving. In this hackathon, each student will have the opportunity to develop a mini project around the hackathon topic, form a team with at least two other students, and compete against others for mentorship, support from established companies, and long term interest from the business community. The applications of hackathon are in the fields of obesity and precision nutrition, innovation and beauty products, and pregnancy. In today's webinar, we will be first introducing the participants to the Microbiome in Health and Wellness Hackathon and provide an overview of Week 1 workshop on microbiome and technology, where we will be covering topics on microbiome sequencing, analytics and bioinformatics, and recommendation system. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. As a part of the hackathon, students will have access to the Omics Logic training that has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. This training has been completed by participants in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. As a part of the hackathon, students will also have access to the Omics Logic resources. But how does it help advance bioinformatics teaching? Omics Logic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omics Logic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large datasets, and introduction to basic coding languages like R and Python, or even beginning a data analysis project. This also comes with an access to the Omics Logic courses, which include courses on bioinformatics, precision medicine, multi-omics, including genomics, transcriptomics, metagenomics, epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of data. This platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of the analysis are performed in different sections on this multi-omics platform. So first of all, let us begin by signing up on the Omics Logic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles. This could be your Facebook, LinkedIn, Instagram, or ID, or even Twitter. A brief bio about yourself that will consist of your educational background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the portal. I'm sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the portal. All right. So here is the link. Right. So for users who do not have an existing account, please click on the create an account option that you see here. Here you will be asked to enter your name, email ID, and provide a password. You also have the option to sign up using any of your social media accounts, which could be Google, Facebook, Apple account, GitHub account, or even Twitter. With that, I'll pause for a couple of minutes for the participants to sign up. Once you have signed up, please put a yes in the chat box so that I know that we are good to proceed. And if you're facing any technical issues for signing up on the portal, you can share that in the chat as well, and our team will be happy to help you. So I'm monitoring the chat box. I'm waiting for the 10 plus participants that we have. All right, so we have one response. Okay, 2 people. If you're having any queries or technical issues, please put them in the chat. We are glad to help you. All right, so since I already have an account on the portal, I'm going to click on the login now option. So this is also applicable for the users who already have an existing account. So once you sign in, you're able to view the various courses and example projects that are applicable here. So for example, if you're interested in metagenomics, you can simply enter the keyword here for metagenomics. And you can see the various courses and example projects that are relevant to this keyword. All right, so now click on the tab that says welcome back with your email ID. This is the part where you will be able to update your account. So for that, click on the icon that says profile. Here you will be able to upload your profile image, add a brief bio about yourself. That could be your education background, research interest. The name should be in appropriate capitalization. And as you can see here, I have linked my LinkedIn account and my Oct-ID. And under the activity tab, the community manager and the mentor will be able to track your learning progress. And the courses that you are currently completing will be appearing under the course tab. So for instance, my interest right now is in Python course, so I'm completing Python course 2. And once you upload any of the projects, that will be visible under the projects tab. And if you're enrolling for any of the programs, for example, I've enrolled in precision medicine program, it'll appear under the programs tab. And once you complete the courses, after completing the quizzes and all of the assignment tasks, you will be able to receive the certificates under the certificates tab. So with that, if anyone have any queries regarding signing up or are facing any technical issues, please put that in the chat and I'll be able to guide you. Otherwise, we're good to go. And now I would like to pass on the stage to Mr. Ilya Brodsky, CEO of Pine Biotech, to give an overview of the hackathon. So over to you.
Thank you, Sri Gauri. That was a great introduction. Welcome everyone to this webinar. I will briefly speak about what this hackathon is about, why we chose to focus on the microbiome, what kinds of opportunities I see for students and researchers, as well as companies that might want to participate. So let's start by briefly talking about why microbiome. I have some stats here on the slide that basically show that startups that focus just on the gut microbiome for health clues raised over 1 billion in VC funding over the past 5 years. This is 2020 and before. So what we see is a tremendous interest in the space from the venture capital community. And that just gives you a perspective on the overall interest in how microbiome can be involved in health, disease, and wellness. Now, we're not going to focus on disease here, but we are going to talk about wellness. So as you can see, it's a hugely growing field and industry. You can see that there are a lot of patent applications that are being applied for. And this has grown almost, I would say, exponentially since the 2010, 2011. And down here, you can see how this interest has a practical direction. So we're not just interested in the microbiome, the basic science of the microbiome, how it works, but we're actually seeing a lot of interest in trying to apply these insights and translate them into some kind of a product or an intervention. So what we decided to do is kind of take a look at data-driven innovation. And what we want to provide the community, the broader community, with are data sets that describe real-world studies and experiments, intuitive solutions, so you can see what kinds of things are people building and what are they doing with this kind of data, and then explain how such data could be analyzed, visualized, and presented in a meaningful way. So for those students and faculty who are interested, we think that this is an opportunity to innovate, solve, and connect. Innovate by being creative, open, and collaborate with others. You can come at these existing solutions that are being addressed by others with an out-of-the-box thinking approach. You can really do something novel. You will also see that this is an opportunity to showcase how you can solve complex problems. And so for a lot of students, this is a way to showcase their abilities, stand out from the crowd, really kind of apply what you have learned, what you know, to a new challenge. And connecting is an important factor here as well. So by working with people who are from the business community, from the research community, from the computer science community, you will see that this interdisciplinary aspect is a huge component of innovation. And we hope that this hackathon could be an opportunity for you to do just that. Now, this is going to be a virtual event, so you don't have to be physically at any single location. You can participate from India, from Africa, from Brazil, from the US, anywhere in the world, really. And I think the benefit of that is because we can see that these trends are going to continue and grow in the future. So not only have we seen work being disrupted, innovation and collaboration has been disrupted as well. And I think that at one side kind of disrupts the way things have been done, but it also provides extreme new opportunities. So if you're in India, you might be thinking, yes, this investment might be happening in the US, how can I actually be involved? But the truth is that more and more venture capital firms in the US are looking outside of the US to find innovation. In the US, you might be thinking, well, how do I do something differently from the way that it's done here, right? And this is an opportunity to find people who might have completely different ideas, different audiences, different targets. Ultimately, this is an opportunity to build and compete. And by building is very different than doing research, right? So we've seen a lot of interest in research problems that result in academic publications. Maybe this is intellectual property, but it is often not a product. And so what we hope to see out of this event are more ideas that have practical applications, or at least a direction at that kind of a solution. What kinds of topics are we going to cover? We're going to focus here on 3 different topics that are all extremely relevant, popular, and interesting in the domain of microbiome. The first one, this is probably something that the majority of the people know about and is the most obvious one, is the obesity and precision nutrition. And what that means is that the gut microbiome has been studied enough to show that there is a critical connection between different types of foods and lifestyles with obesity and precision nutrition. This also has to do with food being a functional medicine. So that means that some conditions, even though they are early on, can become chronic and very complicated to treat with drugs, with surgery, with these severe interventions. But at an earlier stage, they could really be prevented with dieting, with different types of functional foods that help the gut microbiome be restored and function properly. The second, maybe a little bit less known field of application for the microbiome is skin aging and beauty. And we'll actually have an innovator herself and a startup founder herself, but also a researcher, speak about what are some of those innovative and cutting edge applications of the microbiome to the field of beauty and aging. And then the final one is something that all of us have gone through in some way, and this is pregnancy and femtech. So a lot of the microbiome studies have pointed to different associations between, different types of pregnancy that has healthy or more risky outcomes. And so this is a field that definitely has a lot of impact as well. So for obesity and nutrition, we see that the gut microbiome has been studied for associations between diet and the microbiome, healthy weight loss, risk of diabetes, personalized nutrition plans, recommendations on pre and probiotics, as well as different types of sensitivities. And you can see that there are a lot of clinical trials, which means that there's a lot of data sets that are available to test and identify interesting insights from these data sets. So we'll introduce those data sets, and we'll also talk about some of the industry challenges. Now, in the field of the gut microbiome, the obvious challenge is that there are some established companies that come out with very similar tests at pricing that varies between approximately $60 and $200 or more than $200. They use different techniques, and they provide different types of insights, but all of them claim to be the best, the most advanced, the most comprehensive. So the question is, which one is the best, the most comprehensive, the most insightful, and what are some of those use cases that could be turned into innovation that could kind of change the way that things are done today? The skin and microbiome has a lot of applications, from rashes to acne to wrinkles, blemishes, longevity and aging. And so since more and more people are aware of how their skin looks and what they do to make it look better, this has become already an opportunity for people to develop new products. And those new products include things you can buy on Amazon, like Biome is a type of a company that produces a lotion that is matching your skin microbiome. So really interesting direct applications that are already being done in the industry. Now, the question is, how do these things work? And what are some opportunities to introduce new ideas into this space? Now, in the field of femtech, there's also a lot of studies going on for healthy childbirth, immunity and conception, antibiotics, diet and activity, reproductive health. And there are already these products being sold on the market. So I think that there's a lot of interesting associations, even in things like the child's psychology over the years being influenced by childbirth and the mother's microbiome. So there's a lot of interesting ideas here, and I think that this would be an opportunity to both learn about what those things are and try to develop some innovative product. Now, we are going to try to prepare everyone to focus on real problems and real solutions that are possible, that are realistic. And for that, we're going to bring in experts to speak about different types of sequencing technologies. As I mentioned, curated data repositories where you can actually find some of these studies and do something with them. People that specialize in business analysis and framing the problem in business terms, as well as user experience. How do you not only produce an example, but actually turn that into an experience that somebody can use? Why sequencing technologies? Well, sequencing technologies have been one of the main drivers of cost, accessibility, and detail in this field. And so, companies like Illumina and Oxford Nanopore have developed very different approaches, but ultimately now we are in a race to the bottom of the pricing scale, right? So new technologies are becoming much more useful and affordable, and that is driving accessibility for these types of innovations. Now, for all of these different types of sequencing, there are still challenges associated with data analysis. So we will talk about how these data are analyzed, and these include multiple types of omics data, including intuitive and informative analysis types, and that's really where bioinformatics and multi-omics data analysis comes in. So we'll talk about how these types of data are analyzed and integrated. Now, After we talk about just in general, what is the logic for this kind of analysis, we'll actually make sure that everyone has access to easy to use tools that they can produce insights quickly. And we'll make sure that you have access to our T-Bioinfo platform with these different types of pipelines that are available on there already. Now, in addition to that, we'll talk about non-omics data, where to produce a product, you actually have to focus on finding associations between omics and non-omics data, such as maybe different types of lifestyle and activity tracking devices. This is an important topic, and we'll kind of introduce you to how that could be done and what are some of the companies that are doing this already. Now, when we think about this, we're going to look at the business aspect as well. And that means understanding these industry trends that we just spoke about, but also looking at customers and their kind of segments or portions of the customers that might be the best to focus on. And for that, we will bring in investors that will actually speak about what is working and what is not working in this microbiome innovative space that startups are competing for funding in. And they will talk about some previous investments that have been made and whether those have been successful, unsuccessful, and in which way. Now, as I mentioned, we're not going to be the only ones talking. We're actually going to bring in experts in the field that have been working for decades from different aspects, including startup founders, people that specialize in sequencing technologies, investors, existing companies that kind of deal in this space, as well as some researchers that are exploring different opportunities with microbiome data as well. Now, primarily, this event is going to be focused on our partner universities, and that's why we are working to kind of streamline the pipeline. And the main challenge here is preparing students to be ready to address some of these interesting topics and challenges early on, right? So we don't want to spend a lot of time on the basics. We want to jump in to the real opportunities that are there. Now, we have over 10,000 members in this partnership program, and we would love to see your organization join here as well. The community that we've established has grown tremendously, and so we are really excited to see a lot of interest in this space, but we also see the need to continue and train people to be able to use omics data, both for basic and translational research. So with that, I will pass it on to Dr. Raghavendran, who will speak about how this is actually going to work.
Thank you, Ilya. Let me find ways to share my screen and set up my presentation. Can you see my screen?
Yes,
it's thank you very much for the confirmation. So thank you one more time, Ilya. So I'm Raghavendran. I'm working as a bioinformatics mentor with Pine Biotech, and I welcome all of you to this portion of the webinar to introduce you to this hackathon. And let's discuss some basics about in this portion. We will discuss some basics about what is microbiome and what are some of the sequencing technologies that relate omics data analysis with microbiome, and what is the approach in bioinformatics analysis methods towards understanding the association of microbial population with disease. And we discuss this from the point of view or from the popular publications and popular research reviews. So this will actually give you necessary tools and some idea and introduction, and this will prepare you for participating in this hackathon that focuses on microbiome in health and wellness. So let's jump in. Let's start by reviewing a little bit of history of microbiology. So where the study of microorganisms leads to the creation of tools and approaches that are still used today to continue and to uncover the veil of world which we do not see. but yet has such a profound effect on everything that is around us. This all started with, for example, microscopes that helps us observe or that actually helped us observe the first bacterium in 1673 itself. But today we have microscopes that can enlarge from 50 to 300 times the size of the first observation of bacteria that was made long time ago. So as the resolution of these microscopes improved, more and more representatives of this unseen world have been discovered. Today, the microscopes that we are using are tremendously powerful, and it allows us to see smaller and smaller elements that reveal how the world of microorganisms functions on every imaginable surface. So in this example, for example, in this image, we can see a super high resolution image of bacteriophages that are green in color attacking the bacteria, that is E. coli, that is blue in color. So we have advanced to such a level of high resolution, and this actually made us define microbiology not only by the organisms that the research studies or that the field studies, but also by the tools that are used to study them. And these tools, as we already discussed, has evolved to allow us to study and categorize microorganisms specifically. And the technology for bacterial culturing, coupled with the technique to stain bacteria, has historically improved our ability to isolate, to classify, and to determine whether a bacteria is a cause of human disease, or even select appropriate treatment for handling that pathogenic entity. Why are we interested in studying the microorganisms generally? So the connection between disease and microorganisms has been well established since the 1800s. And these studies actually provide important information about bacterial function in isolation. However, as more and more studies came through, it became apparent that most microorganisms live in a complex communities, and many members of which are impossible to culture in isolation. And today, we know that microorganisms, or the microbiota that make up the, for example, human microbiome, are not just unicellular organisms living in isolation alongside each other, but instead they are highly regulated, highly structured, and highly functionally organized communities. So let's take human skin as an example, right? Bacteria are, we know that bacteria are able to grow to almost every surface, forming architecturally complex communities that we call, or that are termed as biofilms. The stability of these biofilms or the communities is fascinating, as many of us probably know from trying to battle common skin conditions such as dry skin, acne, and eczema, for example. So forming these distinct communities, these microorganisms are interconnected and are determined to maintain their community stability. And these bacteria, for example, within the community or from this biofilm, can communicate with each other by producing, detecting, and responding to small signal molecules in a process called quorum sensing, right? This process, or this quorum sensing activities in these communities, are also involved not only in communication, but also in the virulence of the pathogenic bacteria, or in response to, for example, external stimuli, or also in controlling the host defenses, and in responding to antimicrobial agent, etc. So inside such community, or inside each community, there is a balance of bacteria, fungi, and viruses that controls the species abundance and the activity. So this balance is very absolutely necessary and very important for our understanding of the association of this microbial community with not only with the diseases, but also with health and wellness and general well-being of ourselves. So microbes inhabit just about every part of the human body, not only on the skin, right? For example, we already saw some examples of microbes living on the skin, and we definitely see microbes living in the gut and up in the nose and gastroenteric pathway everywhere. The microbes, or the diversity of these organisms that inhabit various human bodies, and are estimated to make up more than half of people's cells, right? And this diversity is extremely critical to human health, right? And most of the time, these microorganisms that live in harmony with with their human host provides extremely important functions that are essential for human survival. For example, from enabling the work of the immune system to underpinning brain function, the microbiome has been linked to nearly every health condition that one can find the association. So today, health-related interventions and well-being related interventions seek to manipulate the population of this microbial diversity, or the diversity of this microbial population rather, through products such as probiotics, through drugs, through therapies, through foods, and through wellness products, and etc., etc. Some of which Ilya already had introduced you. So one such example in this association is detailed in this review, right? This review focuses on the microbial diversity in the gut, altering the responses in the diversity observed in the skin, right? For example, this image actually shows the inflammatory and microbial influences between the gut and skin for a healthy state in the left, and for the dysbiotic state or non-healthy state in the right. And they are compared in detail in this review. So I will shortly summarize it. The dysbiotic state is characterized generally by an impaired gut barrier, for example. This impaired gut barrier results in the imbalance in gut microbiome. And this gut microbiome can be, for example, can also result in reduced mucus layer formation, or reduced immunoglobulin secretion, or this again can promote barrier disruption. This can actually create imbalance in the intestinal permeation into the bloodstream, and also can generally cause gut inflammation. Or this dysbiotic state can also be characterized by impaired skin barrier, right? This generally results in imbalance in the skin microbiome, and this reduces, for example, human and antimicrobial peptide production. This promotes skin rashes, this promotes lesions, skin inflammation, and etc. other conditions. This dysbiosis between the gut and skin are actually connected through a balance between, or balance in the helper T cells or T lymphocytes. And in this dysbiosis, or in this imbalance, this balance is actually skewed towards, this balance is actually extremely skewed, where we can or this review actually focuses on identifying whether this skewness results from the gut dysbiosis or from the skin dysbiosis. So if you want to check, if you want to read a little bit more about this review, let me paste the publications link, if I can get it, in the chat box. Yeah, I got it. So please go through this publication if you are interested in. This is an excellent review that will open your eyes to several different associations, and that can be very, very helpful in general. So moving on, the imbalance, as we saw in one such example, for example, gut microbial diversity, is related to many modulations or many infectious diseases and their pathogenesis. For example, many more studies actually uses the Human Microbiome Project data for the following association, right? The role of gut microbiome has been associated to Crohn's diseases. It is generally found associated with ulcer or esophageal cancer, right? The role of skin microbiome is associated or is generally related to psoriasis, atopic dermatitis, and even sometimes immunodeficiencies. And sometimes in some of the publications, the role of urogenital microbiome is related to reproductive complications, right? And Ilya also focused on the childbirth, right? And several publications and research focuses on understanding number of childhood disorders from gut microbial population, right? Including, for example, pediatric abdominal pain. It also includes intestinal inflammations, and also sometimes a severe condition in which premature infants actually die due to the fact that the intestine actually dies inside them. So these are all some of the interesting starting points that you can explore. So what happens is these researchers and these studies actually fuel this exponential increase in the microbial data or microbiome data that are available in publicly available resources or repositories. And these were significantly advanced through major initiatives in, for example, in the metagenomics of human intestinal tract, or in subsequent human microbiome project. Now we have Earth microbiome project, and a lot of these huge studies that have been made possible through advancements in metagenomic sequencing, right? So the result of this is that not only skin and gut, which are well known to be a carrier of microbial, diverse microbial population, lungs and even brain microbiota are starting to be characterized. And each new project represents extremely vast opportunities and challenges to determine which microorganisms play a key role in the development of certain environmental conditions necessary for healthy functioning of the human organs, or which subset of those microorganisms can be harmful to those human organisms themselves, right? So this actually, as we know, can contribute to critical, this actually can expand our understanding of studying these endogenous human microbial communities that can contribute to critical metabolic and physiological and immunological functions that include maturation of the immune system, that includes, for example, energy generation, metabolic regulation, fat storage, and skin maintenance, and etc., etc. Right? You can explore a lot of these things in this program, in this hackathon. So far we have been discussing about the data or microbial data, metagenomics data, microbial population. So what's in store for the bioinformatician, right? For the bioinformatician's perspective, from the bioinformatician's perspective, metagenomics is the genomic analysis of microbial DNA from environmental communities, right? The tools that will enable us to perform this metagenomics analysis will actually help us understand the population of this microbial community that were previously unculturable or that were previously unexplorable, right? And this ability to identify microbes without an a priori knowledge of what the sample contains is opening new doors in disciplines not only related to, for example, disease biology, but also generally expands the horizons of microbiology, virology, environmental sciences, biomedical research, as such, right? And sequencing-based examinations of metagenomes has actually become a powerful tool for generating novel hypotheses based on the microbial population and microbial community population analysis. Oh, my animation is running slow. Sorry about that. Yeah. So unlike other omics, metagenomics does not study a single organism, right? But a whole community at once. This is a snapshot of collection of organisms directly in their natural environment, right? Since the genomes are so diverse, this is typically accomplished or carried out by sequencing a specific marker portion of the genome instead of all the genomic material present in the given sample. And how are these done? Many of these processes are done using next generation sequencing that focuses either on sequencing, sometimes sequencing the whole genome or transcriptome of the microbial population, or most of the time a popular approach is to select a portion or select a gene of these bacteria or the portions of the genes that are in the variable sections, right? We will discuss this in a little bit while. Yeah, so these can be shortly divided or roughly divided into categorized into shotgun metagenomics or metatranscriptomics approaches and 16S amplicon based approaches, right? The 16S amplicon sequencing is actually a recently developed, it's not now recent, it's very well developed now, technique that is widely used to study microbiome directly in its environment, as we already discussed, right? This approach or this sequencing approach focuses not on the whole or not on sequencing the whole genome of the organism or of the bacterial community, but it focuses on 16S or 18S ribosomal rRNA that can be amplified using several amplification methods. This gene, 16S gene, contains hypervariable regions that can provide species-specific signature that are useful for bacterial taxonomical identification. And this identification can be carried out from a diverse set of these species-specific signature sequences. To account for the diversity of such sequences, they are usually combined into operational taxonomical units or OTUs, which can be used to classify groups of closely related individuals. And OTUs are actually formed from groups of these genomic sequences that represent a cluster or a phylogenetic clade that has that are closely related and that has meaningful abundance in our sample. So by far, the most common type of clustering method for studying microbial studies has been this operational taxonomic units. Right? This works by identifying clusters of closely related sequences with a fixed identity cutoff. However, different regions or different sequences vary in their, for example, sequence identity percentage, and sometimes phylogenetically related taxa share a less predictable identity percentage. This is a limitation in this study, or in other words, this 97% that one uses to cluster these sequences into OTUs might not cover all of the sequences diversity, or might also include some of the closely related species. To address these limitations, in recent years, a denoising method has been introduced through several different popular algorithms as an alternative clustering method. This algorithm actually uses probabilistic method to predict actual sequencing errors, which are noise, before forming clusters. And hence, this clustering method is termed as amplicon sequence variance, and this is based on sequence probability rather than sequence identity. And this results in a fewer but self-consistent custom clusters that have been thoroughly validated to have more acceptable precision in many different publications. So, for example, based on mapping of these specific regions that are amplified from, let's say, 16S rRNA gene in the bacterial ribosome, these OTUs or ASVs can be assigned to belong to particular taxonomical classes of bacteria, and this will actually help us, for example, calculate the abundance of each one of those bacterial family or phyla in the community. And each community can be studied by comparing with the others between similar conditions or different conditions, or between different individuals, or between different state of these individuals, for example, healthy and obese, or generally, people with autoimmune diseases or not, for example. So these analyses will actually include in-depth understanding of data preparation, reference databases, and analytical approaches to, for example, to formulate hypotheses or to answer specific questions based on what we are actually understanding, or what we are actually identifying from these taxonomical classification. So importantly, this sequencing data, for example, will represent only one type of microbial omics, which are becoming many more. Sorry about that. Yeah, this sequencing data will actually represent this metagenomics abundance classification, which will help us in, as we already discussed, in quantifying the abundance levels of different phyla or genus or species level classification of bacteria in the sample. So this is usually done by annotating or mapping these OTUs or ASV sequences to a reference database. And these reference database can actually include a ribosomal database project or repositories such as Green Genes or the Silva database. So these databases contain taxonomical information for the domains of bacteria, archaea, eukarya, based primarily on the phylogenetic information or phylogenes that are built from the small subunit rRNAs, or 16S rRNA for the prokaryotes and 18S rRNA for the eukaryotes. So we can use this, for example, the Silva database to identify and to understand the taxonomical distribution of our OTUs. So what is the next step in this bioinformatics approach towards studying microbial abundance, right? So many questions about the role of microbial members or microbiome members and communities are now being answered using these next generation sequencing genomic data that we can generate. And this also can be done, or this in comparison or in complementary approaches such as mass spectrometry approaches for proteomics and metabolomics. Data generation can also be used to study this or related questions. So the next phase of this microbiome research actually lies in the ability to integrate these multi-omics data sets to combine not only the next generation genomic or metagenomic data, they can also integrate the mass spec data, or they can also integrate the structural data with phenotypical information about the overall changes in the community in their natural environment. So this is where the next few years or next few approaches of biomedical research in metagenomics and microbial population research is taking you to. So as we have discussed about the history of microbiology, the emergence of sequencing technologies, and how this data can be used to study, for example, abundance, microbial abundance in the samples. So let me show you, or let me share with you some of the additional resources that you can rely from the Learn Omics Logic portal to study these topics in greater detail. And this will also include practical analysis tools that you can gain experience to in handling such data sets, right? Let's see. Let it work. Yeah, now it's so. We have courses on metagenomics, and we have example projects that take, for example, that include data from popular publications and explores their effects, or explores the data for their association with what is being studied, or with some novel associations that you can find from existing publications. For example, in Project 19 takes the Rett syndrome data, Rett syndrome microbial population that is estimated for children with Rett syndrome and for healthy individuals, and finds a dysbiosis and relates that dysbiosis generally with the dysbiosis that is characterized, symbiosis and dysbiosis that is characterized after subjecting the patients and population to variety of different antibacterial or antibiotic treatments. So that's an excellent project, for example, to integrate information from more than one source of publication. And we have several other projects, such as projects that focuses on human microbiome data, and a project that focuses on model system, like a mouse model system, so which we will shortly discuss. So let's talk a little bit about the approaches that you will be introduced to, and throughout this course you will be given access and directions to the server tBioInfo platform for bioinformatics processing and for analyzing these metagenomics data. This platform includes demo pipelines, and this also includes analysis approaches to run metagenomics bioinformatics pipeline. Different stages of this analysis are performed on different, different stages of this analysis are performed on different sections of this multi-omics platform. So you can explore not only on this metagenomics area, but also the other ones when you are interested to do. So the interface of this server will enable you to quickly build logical graphs of analysis methods and sequence pipelines to understand each steps and their significance in this analysis. For interested people, the implemented pipeline can be accessed using the demo pipeline that I will show you how to do that. So let's quickly go through, for example, a demo pipeline of a project that we will definitely get our hands on in this program. So this project is a practical example of understanding the role of high-fat diet on obesity, right? So this is the project number 13 in the Learn Omics Logic portal. So when you have, if you have the subscription, or when you get your subscription, you can get introduced to this project. This is an extremely interesting project where, oh wait, what happened? Yeah, so where the aim of the study is to test the hypothesis that the obesity associated microbiome undermines behavior even in the absence of obesity. Okay, something happened. Can you see my screen still? I'm getting screen not shared.
Yes, we are able to see
Yes,
the
sir, screen is
Okay,
green.
sorry. Thank you. Yeah, I'm getting screen is not shared. Okay. Okay, let's see. So for example, in this study, generally, the mouse will actually receive the microbial population that is being generated or that has been extracted from mouse that are fed with normal diet and mouse that are fed with high fat diet, and we will see associations of the changes in these microbe in these mouse and their behavior, and how the abundance levels of these microbial population addresses relates to the dietary supplements that we can associate with the respective mouse. Let me reshare my screen again to take you to the demo pipeline that I was talking to you about. To reshare. Okay, I got it. Okay. Okay, can you see my screen? Are it still sharing?
No, it's not sharing now.
Yeah, it's not sharing now. I also got this. Where is this? Okay, I think you should be able to see it. Can you see my screen? Can you confirm what are you actually?
Yeah. We're able to see T bioinfo areas for areas of analysis.
Perfect. Thank you very much. So this is a project that I was talking to you about. This project is an excellent starting point where the use of the model system will actually ease you into the metagenomics analysis, where we do not see complications due to the sample diversity or, for example, which you will generally observe when you acquire sample from human systems or real life human systems. So this is an excellent project to start with. And in this project, after introducing you to what this study is about and what is the background and how are we going to set up a hypothesis to study their association, then we have a practical hands-on of performing these 16S amplicon sequencing analysis in the Turbo Bioinfo platform. So this is explained to you in lesson number 5 that we will go through using the tBioinfo platform server. So in this server page, if you come down, scroll down, in the metagenomics, under the metagenomics section with amplicon 16S and 18S analysis, then you can drop it down to access the demo pipeline, that is Chow versus HFT. So if you have access or if you have registration credentials to server, you can access this demo pipeline. We have prepared this demo pipeline for you to get a taste or get an understanding of how to set up this analysis pipeline for the data or for the input files that we have already uploaded to this necessary pipeline analysis. So anybody who has login credentials to this server can access this demo pipeline irrespective of their subscription status. So what do? You start. So where do you go next is highlighted with the yellow highlight, and in each and every one of these steps, what is being done is actually described here in brief. So we perform some pre-processing of our data. So interested people can read through this to what has been actually done. And for example, in this will have links to their respective methods and citations for the respective methods and publications that talk about those analysis. So each one of these steps, you will get highlight for possible options or possible sequence analysis approaches that you can add next. So here, to guide you through these analysis methods, only the next step is highlighted. So in true, when you are going to input your data, you will be presented with multitude of different pathways that one can take. So you have to build the analysis pipeline depending upon the kind of question that you are asking and the data that we are using for analysis method. So once this is done, and then once the end is done, you will get access to all the output files. That includes all the plots, all the tables, and etc., and also accessible interactive plots that you will get your hands on to. Right? You'll get abundance plot, and you'll get relative abundance plot, where you can understand. For example, if you check the abundance plot, you will see that this bacterial phyla, or I think this is grouped by bacterial, I hope it is phyla, I forgot. So Rickenelaceae, right? If I can pronounce correctly, right? It looks like this Rickenelaceae has a different abundance level for different sample types. So this sample has a lot of them, and this sample has a very small quantity, and this sample has even more higher quantity. So just plotting this in their absolute value is not very informative. So when we use the proportional abundance plot, then we can clearly see the pattern that in proportion to the total number of abundance that we observe for that particular sample, and these three samples have a very comparable amount of this specific species, while these three samples have a lower abundance level of these species. So this is how we can interpret the data, and more on this level of interpretation and even more methods of interpretation and usefulness of this tool. We also have an interactive option, which is not loading right now, but when you get your access to this server, you will see that loaded up here, which is a dashboard that you can change the parameters that govern these abundance calculation. For example, if I want to change it from phyla level to species level, that you can do it there. And if I want to change it from species level to genus level to see this grouping, I can quickly change it there without running this analysis all over again. So this, and also more downstream analysis on, for example, platforms, for those who are interested on platforms like in R and R in Python, are actually given in this lesson. You can definitely check that out. So with that, I want to pass the stage back to Sparsh, I think.
Yes, now
Yes.
we can pass it on to Sparsh for the hackathon event details, such as registration process and so on. So over to you, Sparsh. Sparsh, are you able to?
Yeah.
Okay, sure.
I'm so sorry. Yeah. So I was unable to unmute myself. And now, like, can you hear me? I am audible?
Yes, you're audible.
Perfect. So thank you, Dr. Raghavendran, for the wonderful explanation. And I'll quickly make you through, like, tell you all about the registration part of Hackathon. So I'll just share my screen. Give me a second. Could you please confirm, Shri Gauri, if my screen is visible?
Yes, we can see the hackathon language. Yes.
Perfect. So this is the landing page of Hackathon, and I'll share the link with you all in the chat box itself. So after the, okay, this is something else, sorry. So this is the landing page. I'll share the link in the chat box itself so that after the meet, you can go through by yourself and see what exactly we are including in the hackathon, what kind of workshops we are doing before workshop before actually like going into the hackathon, etc. So there are like the first 3 weeks are devoted to different kind of workshop. So this particular webinar was devoted to tell you, to brief you, to give you an insight about the week 1 workshop, which will be starting from November 1. So it was related to microbiome and technology. We covered here microbiome sequencing, analyticals and bioinformatics and recommendation system, and how you can leverage T-Bioinfo server and other resources on our Learn portal and get more into and learn more about microbiome sequencing, metagenomic sequencing, etc. So the next webinar is next week, and we'll be inviting you, and we'll be sending you calendar invite and invitation mail regarding the week 2, regarding the second webinar, which will be devoted to the human microbiome. And we'll be telling you and giving you an insight of what we'll be including in the second week of the workshop here. And same goes for the week 3 workshop. Now moving further, so these are some of the like experts, commercial and academic experts, who will be there in those workshops, and they'll be guiding you throughout the hackathon, what you need to do so as to so that you can make a kind of a MVP and present it further. Now moving further to the registration cost for the hackathon. So there are 3 registration costs for the hackathon depending upon like your participation. So for an individual student, as we are a US based company, in the beginning itself, Sri Gauri explained that we are a US based company, so the pricing is in dollars. So for the individual student who needs, like, who is willing to participate for the hackathon, the registration cost is $100. For a group of students, so by a group of students we mean that 4 to 5 students, the registration cost is $300. And for the partner university, so here partner university means those universities with whom we have collaborated, with whom we have signed a MOU. So the costing is $100. Now we have provided the partner university with a 65% of discount to those universities who are like partnered with us and collaborated with us. Now if you'll move further, you will see 3 tabs or 3 buttons. These 3 buttons are obviously for these 3 different registrations. One is for individual student payment gateway checkout, the other is for group students for 3 to 4, then the third is for payment university, partner university payment gateway checkout. So those who are either from a partner university or a group of students from any other university or an individual student can accordingly choose the payment gateway, and they can directly click on this button and it will be redirected to the payment page. So they can fill out the credentials, like the details, the payment information, and they can um, submit from here on. And like, we'll get the submission and we'll be sending you the further details accordingly, that about the November 1, the week 1 workshop, then the second week 2 workshop, then the third week 3 workshop, and the finally November 23rd to 28th, where we'll be doing the presentation of the minimal value product which you have created, your team has created together. So this was about the registration and the cost of the hackathon. Obviously, in this particular cost, we will be providing you with some resources. We'll be providing you the access on our Learn portal, the access on our T-Bioinfo server, and many other resources. So this was all about the registration cost for the hackathon for now. And if anyone is having any kind of doubt regarding the registration, the cost, or the grouping of the students also, so please feel free to ping in the chat box itself, and I'll be there to answer your queries. Thank you.
Yes, Parsh. So we have a query from Dara, that is, can we join a group?
Yeah. So definitely you can join as a group. So here it is what like we have mentioned. So in a group there could be 3 to 4 students, and the cost is $300 for them. So the students can be a mix and match fields or domains. A student can be of a genomics background and a computer science or a MBA, or whosoever like who's having expertise in these all fields can, they all can group together and like participate in hackathon and come up with the MVP. So surely we can, like, you can join a group.
So Stephen is asking, by a week, how many hours should one at least dedicate for the course per week?
So it depends upon you, Steven. So we'll be providing you with like there will be live kind of sessions also, where we'll be inviting the commercial and the academic experts from different domains and areas. So there will be some live sessions where we'll be also providing you with some kind of data, and that would be asynchronous. So we'll be mailing you out with online associated resources, and you can, according to your time availability and feasibility, you can go through with that resources and complete. So it totally depends upon you, maybe 2 hours a day, or it depends upon you. So yeah,
All
I
right.
hope.
So thank you so much, Sparsh. So do we have any further queries? If so, please put them in the chat box. Otherwise, you could also reach out to Sparsh via the email ID that I've shared in the chat box. You can also reach out to her via her contact number, which is also active on WhatsApp. So with that, thank you all for joining today's webinar. I hope to see you all. All right, so we have another query. So up to how long will the server be accessible?
So for 1 month, we'll be providing you with the access of the server.
And Sarah is asking, is there any WhatsApp group as well?
So yes, Sarah, there is a WhatsApp group. Like we have a bioinformatics community called Genomics. We have a club, and we on daily basis update there about our workshops, about our webinars, or what we are conducting next. So what I'll be doing is like I'll be sharing the video recording at this particular session, and I'll be including in the mail itself the WhatsApp link. So you can see in that particular mail, and you can join from there as well. Or if Ojasvi or any other group member is here available, they can share the link of WhatsApp group. That would be great. Otherwise, I'll mail you.
Yes, so or just we will be sharing the link to the WhatsApp group so everyone interested can join. So do we have any further queries in the meantime about the program registration or the team participation? If so, please put them in the chat. We'll be happy to help you. All right, like Sparsh had mentioned, all right. Okay, thank you so much, Nilaba. So we have another webinar in next week. So we'll be mailing out the details of that as well. It will be about the workshop 2 of the hackathon. So I hope to see you all for that webinar as well. And I hope to see you all participating in the hackathon. So I would highly urge all of you to make the most out of such a wonderful opportunity. Stephen is asking, is this recording going to be shared? Yes, it will be shared to you via your email. Thank you very much, Stephen. All right, so Ojasvi has shared the link to the WhatsApp group as well. So those of you who are interested to know more about the bioinformatics opportunities and get up-to-date news about bioinformatics happenings, then you can join the group. So with that, thank you everyone for joining today's webinar. I hope to see you all next week as well. And with that, thank you everyone. Have a great day. Thank you.